Going to start with some basic high-yield MCQs in relation to the topics GIT and liver, respiratory system and renal system. Okay. So these are some of the high-yield topics in relation to GIT and liver. Jaundice, its type and cause are important. Deglutition, peristalsis, different glands, their secretions and functions, especially the gastric glands. Okay. So let's start with the MCQs. So the first one is, in jaundice, there is an unconjugated hyperbilirubinemia, which is most likely due to. So the options are hepatitis, cirrhosis, obstruction of the bile canaliculi, and increased breakdown of red cells. So basically, they are asking about different types of jaundice, not different type of jaundice. One of the jaundice called prehepatic, hepatic, and the obstructive or the post-hepatic types are the different types of jaundice. Okay. So here the answer here is in jaundice, there is an unconjugated hyperbilirubinemia, which is most likely due to the options are hepatitis, cirrhosis, obstruction of bile canaliculi, and increased breakdown of red cells. So the answer here is increased breakdown of red cells. Okay, so there are different types of jaundice. The first one is the prehepatic, and second is the hepatic, and third is the post-hepatic or the obstructive type of jaundice. Okay, so here they are asking about the unconjugated hyperbilirubinemia. Okay, in these different types of jaundice, the bilirubin that's elevated is either it can be unconjugated, it can be conjugated, or the mixture of both. Okay, so let's study about what the jaundice is. Jaundice is basically it's a yellowish pigmentation of the skin and mucous membrane and deeper tissues due to excess bilirubin. Okay, and here in hemolytic jaundice, what happens is it's a prehepatic type of jaundice. The liver function here is essentially the normal. Whereas in the obstructive type, liver function is exaggerated. And in hepatic type, liver function is altered or damaged. Okay. So these are the different types of jaundice. First is about the prehepatic jaundice. In this type of jaundice, what happens is excessive amount of bilirubin is presented to the liver, which itself can conjugate. I.e. normally liver will conjugate the normal amounts of bilirubin which are present in the body. But here the amount of bilirubin is so much that liver cannot handle it. So the type of bilirubin that's elevated is unconjugated. Okay. That's why prehepatic type of jaundice, elevated unconjugated bilirubin is seen in the serum. Okay. Next type is the hepatic type. In hepatic type, as the name indicates, the dysfunction is of the liver. Okay. So impaired cellular uptake, defective conjugation or abnormal secretion of the bilirubin by the liver cells. So here what happens is both conjugated as well as unconjugated type of bilirubin is elevated in the serum. Okay. In the first type or the prehepatic type, which are disease that's causing the excessive breakdown of RBCs that will cause the prehepatic type of jaundice leading to unconjugated hyperbilirubinemia. Okay, in the post-hepatic type, here the liver cells are normal, but due to the impaired excretion due to the mechanical obstruction to the bile flow. Okay, some obstruction is happening in the biliary tract that will lead to excessive conjugated bilirubin in the serum, because what happens is liver can conjugate. Liver is normal essentially. But what happens is due to obstruction, the conjugated bilirubin is settled in the liver itself and it's stored in the gallbladder, and hence the serum, it is the elevated type of conjugated type of bilirubin is elevated in the serum. Okay. So another two syndromes which are in which the unconjugated bilirubin is elevated is Crigler-Najjar syndrome and Gilbert syndrome. Okay, Crigler-Najjar syndrome, that's autosomal recessive type is there, another autosomal dominant type is there, and one is very fatal form and it's usually present at birth. Okay, Gilbert syndrome is almost somewhat milder variety of this unconjugated hyperbilirubinemia. Okay, the patient has a good prognosis in case of Gilbert syndrome. So this is also another PYQ in which the bilirubin is conjugated in liver with glucuronic acid. So liver conjugates the bilirubin with glucuronic acid. Okay. So another question is urobilinogen is absent in which type of jaundice? Okay. The options are obstructive jaundice, viral hepatitis, hemolytic jaundice, all of the above. So what's the answer here? Urobilinogen is absent in obstructive jaundice. Okay, I will explain why it's elevated in obstructive type of jaundice. Okay, So why it is elevated in the obstructive type of jaundice? Obstructive means what? Some obstruction is happening in the biliary tract. Normally, the common bile duct along with the uh, pancreatic duct joins the ampulla of Vater or common hepatopancreatic duct, which opens into the duodenum. Okay. So what happens is it's released into the intestine. And in the intestine, what happens is the conjugated bilirubin, about 50% of the conjugated bilirubin, it returns to the liver itself, and some amount of urobilinogen, which is present in the intestine, that's acted upon by the bacteria. Okay. Sorry, conjugated bilirubin that's present in the intestine is acted upon by the bacteria, about 5%. It is converted to urobilinogen, which is excreted from the kidneys. And some amount is also excreted via the feces. Okay. In obstructive jaundice, what happens is this conjugated bilirubin is not reaching the intestine, and hence it is not acted upon by the bacteria, and hence urobilinogen in no way appears in the urine. Okay, because of the obstruction. That's why in obstructive jaundice, urine urobilinogen is absent. Okay, because this conjugated bilirubin is not reaching the intestine because of the blockade in some part of the biliary system. Okay, that's why in obstructive jaundice, urine urobilinogen is absent. Okay, coming to the next question. The most important function of hydrochloric acid in stomach is? The options are destruction of bacteria, neutralization of chyme, activation of pepsinogen, stimulation of pancreatic secretion. So what's the answer here? So the answer here is activation of pepsinogen. Destruction of bacteria is also a function of hydrochloric acid, but they are asking about the most important function, and hence the answer becomes activation of pepsinogen. Now, what are the functions of HCl in stomach? That's the first function is the basic or the most important function is the activation of pepsinogen to pepsin by providing an acid medium. And this pepsinogen is produced from the gastric glands, usually by the chief cells. So chief cells of the gastric glands produce the pepsinogen. Pepsinogen is secreted in an inactive form, which is converted to pepsin, its activated form. This occurs only in low pH or the acidic pH. Okay. To provide this acidic pH, this HCl is of help. And the other actions of HCl is the bacteriolytic action, also the acidification of the chyme, which cause the release of secretin and the CCK hormones. Okay, cholecystokinin hormones. Okay, so secretin is an inhibitory hormone. Okay, so when the chyme reaches from the gastric glands after the digestion from the stomach, it reaches the intestine. So gastric secretion has to be stopped after this chyme reaches the intestine, so that this secretin is secreted from the small intestine cells. Okay. So the main duct of pancreas is always called Wirsung's duct. Okay, Wirsung's duct. And this is also the same question in a different manner. Pepsinogen is activated by, here they are asking about the pH. Okay, so low pH or the acidic pH is the answer here. Now coming to the next question. Intrinsic factor which helps in the absorption of vitamin B12 is produced by. Okay, the options are parietal cells of stomach, chief cells of stomach, beta cells of pancreas, goblet cells. So what's the answer here? It's the parietal cells. Okay, so these are the secretions from different glands of stomach. Okay, stomach is divided according to the location into fundic glands, pyloric glands, and the cardiac glands. Okay, from the fundic glands, there are different types of cells called chief cells, which produce pepsinogen and renin. Okay, chief drinks the Pepsi. Okay, like that you can remember. And the parietal or oxyntic cells says high. That means HCl and intrinsic factors secreted from the parietal or the oxyntic cells. And the mucous cells, they basically secrete the mucus. Next is the pyloric glands. They secrete the gastrin. Okay. G cells. Usually G cells secrete gastrin. And the cardiac glands, they secrete mucus, and small quantities of pepsinogen is also secreted from the cardiac glands. This also you have to remember, because in multiple choices or different choices, sometimes A and B, they might give you the question like this. what pepsinogen is secreted from the chief cells, and cardiac glands are there, and the options have A and B. You can select the best option, that's A and B. Okay, some amount of pepsinogen is also secreted from the gastric glands. Okay, so another type of cells are seen in the cardiac region, that's a endochromaffin cells or the Kulchinsky cells, which produce what? Serotonin. And endochromaffin-like cells are also present. They produce the histamine. Okay. And renin. Renin here is renin. Renin is secreted. Single N is secreted from juxtaglomerular kidney cells, which sends the changes in the renal perfusion pressure via the stretch receptors in the vascular walls. Okay. Chief cells are producing renin, whereas JG is producing renin. Okay. So, another question is that maximum body secretions is produced by the gastric glands. That's also a PYQ. Next question. This is also a very important topic. What are the phases of deglutition and what are the important events in each phase of the deglutition? Okay. So, what the question is, pharyngeal phase of deglutition, the options are, is a reflex mechanism, vocal cords are closed, is a voluntary mechanism, and the fourth option is A and B. So, what's the answer here? A and B. That is, it's a reflex mechanism, as also the vocal cords are closed during the pharyngeal phase of deglutition. Okay. So, deglutition has three stages. First is the oral stage, second is the pharyngeal stage, and third is the esophageal stage. Only the oral stage is voluntary in nature. Rest others, that the pharyngeal and esophageal stages, they are involuntary. Okay, the hypoglossal and the glossopharyngeal nerves are basically concerned with the oral stage. Oral and the pharyngeal stages of deglutition, and the vagus nerve is concerned with the esophageal or the third stage of deglutition. So, basically what is deglutition? It's the act of, the options are peristalsis, incising, chewing, swallowing. Answer is swallowing. So, deglutition is the act of swallowing. It's a reflex mechanism. Mastication is also a reflex mechanism. Okay. So, coming to the next question, that's also about the deglutition. The stages of deglutition consist of voluntary and involuntary. As I've already mentioned, voluntary stage is the oral stage. Which of the following is responsible for small amount of bolus being entering in the esophagus? So, the options are soft palate, palatopharyngeal arch, esophageal sphincter, larynx. So the answer here is esophageal sphincter. Okay, I will explain why. So, deglutition has three phases. First is the voluntary phase or the oral phase. Second is the pharyngeal phase and esophageal phase. And the autonomic nervous system controls the latter two phases and they are involuntary. So, sympathetic and parasympathetic influences are there for the pharyngeal and the esophageal phases. And they are involuntary also under the nervous control or the higher system control. Okay. So, first phase is called oral or the buccal phase. There is a control in the face and chewing is completed and swallowing is set into motion. Okay. By the actions of the tongue. Usually, posterior part of the oral cavity demonstrates a positive pressure which pushes the bolus into the pharynx. Okay. That's what the voluntary phase or the oral phase is. So, in oral phase, what happens is the food from the mouth or the bolus from the mouth is essentially carried to the pharynx. And after that, from pharynx to esophagus, it is done in the involuntary, that is the pharyngeal phase. From esophagus to stomach is why esophageal phase. Okay. So, tongue usually moves upward and backward against the palate, pushing the bolus to the back of the posterior part of the oral cavity and into the oropharynx. I.e., a positive pressure is created in the posterior part of the oral cavity by the movements of the tongue, which pushes the bolus towards the pharynx, and other muscles which keep the mouth closed and prevent the food from falling out. Okay. So, this one thing is called an important thing, that is the bolus is placed over the posterodorsal surface of the tongue. That is what we call as preparatory position, which is the first step of this voluntary or the oral phase. Okay. Next is the pharyngeal phase. This is a very important phase. This is the most important phase in this deglutition. And pharyngeal swallow is a rapid sequential activity occurring in a Second, it's an involuntary activity. It has two things to be noted. First is the food passage propelling the food bolus through the pharynx and upper esophageal sphincter or UES to the esophagus. And second is the airway protection, i.e. this area is common pathway for the air as well as the food. So it's important to make the food pass through the pathway which is made for the food itself and not towards the airway. Okay. So in pharyngeal phase, what happens is the stimulation of receptors in the oropharynx. It sends impulses to the deglutition center. In the last session, we have studied that the deglutition center or the swallowing center is present in the medulla oblongata. So in pharyngeal phase, receptors in the oropharynx, it sends its impulses towards the deglutition center that's located in the medulla oblongata. And impulses are then sent back to the uvula and soft palate, causing them to move upward and close of the nasopharynx in order to prevent the food entry backwards into the nasopharynx. Okay. And the laryngeal muscles also constrict to prevent the aspiration of food into the trachea also. And the contractions of the pharyngeal constrictor muscles moves the food bolus through the oropharynx and laryngopharynx. And also the upper esophageal sphincter relaxation allows the food to enter the esophagus. Okay. So this is basically the pathway of food from mouth to esophagus. Sorry, mouth to pharynx and then to esophagus. Okay. I will show you in the next diagram and explain. So, opening of upper esophageal sphincter. It is formed by the cricopharyngeus muscle. Upper esophageal sphincter is formed by the cricopharyngeus muscle and it's essential for the bolus entry into the esophagus. Okay. So, another important thing that's happening in the pharyngeal phase is that in order to prevent the food, I will explain using the diagram. Okay. So this is the diagram. Initially, what happens is that the bolus of food is in the oral cavity, and after its entry into pharynx, it can go to four pathways. It can have four pathways. First is upward towards the nasopharynx, it can go. Or it can move backward into the oral cavity. Or it can move forward into the larynx. Okay. So in order to prevent these motions and to guide the food towards the esophagus, there are different mechanisms. Protection mechanisms are there. Okay, the position of the tongue as well as the positive pressure that's seen in the posterior part of the mouth prevents the food entry back into the oral cavity. Okay, the upward movement of the food into the nasopharynx that's prevented by the extension of the soft palate, that is uvula, moving backwards and close the pathway towards the nasopharynx, thereby preventing the bolus entering the nasopharynx. Okay. And towards the larynx, what happens is, we can see the epiglottis. So the backward motion of the epiglottis as well as the upward and forward movement. This is the upward and forward movement of the pharynx. Prevents the food from entering the, larynx. That's the upward and forward movement of the larynx prevents the entry of bolus into the larynx. Okay. Also, this larynx by its way, it pulls the epiglottis back from the pathway towards the esophagus. Okay. Also, it will close the vocal cords. Vocal cords is also closed. Approximation of the vocal cords also occur in order to prevent the entry of food into the larynx. And another thing is that momentary cessation of the breathing also happens during this swallowing. That's what we call as deglutition apnea. So these are the important things that's to be remembered in the case of pharyngeal phase of deglutition. Okay, now coming back to the previous slide. So here we have mentioned all these things. Okay. Next is the third stage or the esophageal stage. In esophageal stage, what happens is that food is entering from the esophagus towards the stomach. So what is esophagus? It's a tubular structure from the lower part of the upper esophageal sphincter. Upper 3 to 4 centimeter of the upper esophagus is made into a sphincter. It is called upper esophageal sphincter, that is made up of cricopharyngeus muscle to the lower esophageal sphincter. That's the location of this esophagus. And the lower esophageal sphincter is also tensioned at rest. Okay, during the esophageal stage, after entering the stomach, what happens is LES or the lower esophageal sphincter should be closed to prevent the regurgitation of food and digestive juices into the esophagus and damaging the esophagus. The lower esophageal sphincter should be closed after the esophageal phase or the food after reaching the stomach. Okay, it relaxes during a swallow and allows the bolus to pass to the stomach. For the passage of food from the esophagus to stomach, it has to relax. That is, a lower esophageal sphincter has to relax, and after that, the cervical esophagus or the upper one third, that's a upper esophageal sphincter. It's mainly composed of striated muscle, but the thoracic or the lower two-thirds is made up of smooth muscles. So peristalsis can usually occur in the lower two-thirds. Although this upper esophageal sphincter is also involved in the peristalsis, that is called primary peristalsis. And after the rest of the parts, they are composed of smooth muscles and they are involved in the secondary peristaltic movements. Okay. And the bolus transport to the thoracic esophagus is quite different from that of pharynx because it's a true peristalsis regulated by the autonomic nervous system. Okay. That is the upper part. It's made up of striated muscle and hence it's called primary peristalsis. And after that, lower two-thirds, they are made of the smooth muscles and hence they are performing the secondary or the true peristalsis that is regulated by the autonomic nervous system. And once the food bolus enters the esophagus, passing the US, the peristaltic wave carries the bolus down to the stomach through the lower esophageal sphincter. And usually the peristaltic wave that consists of two main parts, an initial wave of relaxation that accommodates a bolus, and it is followed by a wave of contraction that leads to its propulsion. And gravity usually assists the peristalsis in upright position. When we are eating in the upright position, gravity also helps the propagation of the bolus. Okay. So, another question. Peristalsis in the gut is due to. The options are periperistaltic intestinal secretion, mechanical distension, simultaneous action of circular and longitudinal muscles. Both is the extrinsic nervous influence. So why it is due to mechanical distension. Okay, that is the upper part relax and accommodates the bolus, and after that a wave of contraction that will propel the bolus. Also, the movement of food in the interdigestive phase through the gastrointestinal tract is done via. The options are peristalsis, segmental movements, continuous movements, intermediate movements. The answer is peristalsis. Okay. So this I have already explained. Coming to next question. Peristalsis involves a coordinated contraction and relaxation. The options are above and below the foot bolus, lateral and medial to foot bolus, dorsal and ventral to foot bolus, none of the above. Answer is above and below the foot bolus. Okay. Above, there is a wave of relaxation which accommodates the bolus, and after that, in order to propel it, there is a contraction wave. Okay. Another question is, small intestinal peristalsis, it is controlled by the lower parts of the intestine, lower part of the esophagus as well as the intestinal movements, they are controlled by the autonomic nervous system. Basically, the part of the autonomic nervous system that is situated in the GIT, which is called meyenteric plexus, okay, or the Orbach plexus, that we will discuss in another question. So what is peristalsis? It's an inherent property of many syncytial smooth muscle tubules, and stimulation at any point in the gut can cause a contractile ring to appear in a circular muscle. So inner circular muscles and outward there are longitudinal muscles. Okay, this two muscles help in the propulsion of food through the GIT, and this ring then spreads through the gut tube. And the basic propulsive movement of the GIT is called peristalsis. So this is the movements upward and above and below. Okay. So not only it is seen in the intestine, but also it can be, this peristalsis can be noted in the bile ducts, glandular ducts, ureters, and many other smooth muscle tubules. Usually smooth muscles are involved in peristalsis. Okay. So these are the basic intestinal movements. In small intestine, the propulsive movement, it propels the chyme forward. That's what we call as peristalsis. Okay, and also there is a mixing movement which is called segmentation in case of small intestine. Whereas in large intestine, the propulsive movement is called mass movement, large mass. Okay. It's in the large intestine, so we can remember it like it's a mass movement because it's large. And the mixing movement is called ostration. Okay. So what is segmentation or the mixing movement? It is the back and forth action that breaks apart the chunks of food, or the food bolus is split into small pieces, and it is mixed with the digestive juices. So why this is split into multiple pieces? In order to ensure the maximal absorption of the nutrients by the epithelial cells. Okay. So like this, the bolus is passing and it is split into multiple pieces in order to aid the absorption as well as the mixing of the bolus with the digestive juices. Next is the mass movement that's involving the large intestine. And it is also known as the giant migratory contractions and includes a pattern of motility that is characterized by very intense and prolonged peristaltic contraction, which strips an area of large intestine clear of contents. Okay. So it's an intense and prolonged peristaltic contraction. So a large amount of intestine is cleared, okay, during this mass movement. Next is hostration. So hostra, it means saccules, okay, in the colon. That is given, that name is given due to its segmented appearance. Okay, and hostral contraction, it is activated by the presence of chyme, and it serves to move the food slowly into the next hostra or the saccule, along with mixing the chyme to help in water absorption. Okay, so food is moving through different hostras or saccules. Like this. These are the hostrums of different saccules present in the intestine. That's the large intestine. Okay. So coming to next thing, that is the cephalic phase of gastric secretion is mediated by. So gastric secretions has basically three phases. Okay. First is the cephalic phase, and it's mediated by the vagus nerve. Usually it is neuronal control. It's having a neuronal control. There are two reflexes are there. Unconditioned and conditioned reflexes are involved in the cephalic phase. Okay. Cephalic phase means something related to the stimuli arising from the head region. Okay. So the cephalic phase is purely under the nervous control or the vagal nerve control. And gastric phase, this is under the nervous as well as the hormonal control. And the intestinal phase, it's also the control of various gastrointestinal hormones. Also, some amount of neuronal control is present in the intestinal phase also. Although its majority is controlled by the gastrointestinal hormones like gastrin, androgesterone, VIP, GIP, etc. Okay. So, there are different experiments which can demonstrate the cephalic phase of gastric secretion. Okay, here the question is asking about the cephalic phase of gastric secretion can be demonstrated by the following experiment. The options are Hayden-Haynes pouch, conditioned reflex, Pavlov's pouch, none of the above. We have already studied in our school time also, this cephalic phase or the conditioned reflex is demonstrated by Pavlov's experiment. Okay. So the answer here is Pavlov's pouch. So these are the different experiments. And Pavlov's pouch is used to demonstrate the cephalic phase of gastric secretion. And Hayden-Haynes pouch, it demonstrates the role of sympathetic and hormonal regulation of gastric secretion after vagotomy. Okay. And Bickel's pouch, it demonstrates the role of hormones in gastric secretions. And Farrell and IV pouch, it demonstrates the role of hormones during gastric and intestinal phases of gastric secretion. And another fourth phase called interdigestive phase is present in human beings, which are usually, it denotes some amount of gastric secretion is occurring during, in between meals or during the fasting time. That's what we call as interdigestive phase. Okay, and in order to demonstrate the interdigestive phase, we are using the Farrell and IV pouch experiment. Okay. So all of the following causes the secretion of gastric juice during the cephalic phase except. The options are food in the mouth, sight of food, food in stomach, thought of food. What's the answer? Here the answer is food in the stomach. That is, cephalic phase is the first stage of gastric secretion. It has unconditioned and conditioned reflex. Unconditioned means the presence of food in the mouth. It stimulates the salivary secretion. The taste buds are stimulated. The gastric secretions or the HCl, etc. is stimulated. That is what we call the unconditioned reflex in the cephalic phase. Whereas conditioned reflex means though the presence of food is not an essentiality, only the sight, thought, and the smell of the food can initiate the secretions. That is what we call as unconditioned reflex in gastric phase. Okay, so here they are asking this sight of food stimulates the secretions, thought of food stimulates the secretions, food in the mouth also secretes. That's the conditioned reflex. Sorry, unconditioned reflex, and this is the conditioned reflex. But food in the stomach, food is already present in the stomach. That's not leading to cephalic phase. Okay, that's the next stages. So secretion of gastric juice basically occurs in the cephalic, gastric, and intestinal phases. So cephalic phase is only purely neuronal control. The secretion of gastric juice by the stimuli arising from the head region, that's the cephalic region. That's sight, smell, thought, hearing about the food is called cephalic phase. And the gastric secretion occurs even without the food in mouth. That's what we call as conditioned reflex. Okay. So taste, that is in the food in the mouth, leads to activation of the taste buds, salivary secretion, etc. It is caused by the stimulation of vagus, and it will lead to secretion of the gastric juices. So this gastric juice that is secreted during the cephalic phase is called appetite juice. It is very minute in quantity, but it is rich in enzymes and hydrochloric acid. Next is the gastric phase, that is controlled by both the hormonal control as well as the neuronal control is there in the gastric phase. The secretion of the gastric juice when the food enters the stomach is called gastric phase. Okay, so what happens is the bolus reaches the stomach, and it will lead to the stimulation of vagus nerves, local nerves like mandric plexus, and gastrin secretions. It will lead to the stimulation and secretion of the gastric juice. Next phase is the intestinal phase. It's almost purely hormonal, although some amount of neuronal control is also present. Okay. So intestinal phase is a period of secretion of gastric juice when the chyme enters the intestine. Then the name bolus changes to chyme. Okay. And the chyme in intestine that is stimulated by the gastrin, leading to stimulation and secretion of, in the early stage of intestinal phase, there is increase in, due to the secretion of gastrin from the small intestine. What happens is some gastric secretions, so the amount of gastric secretions is increased in the initial stage of intestinal phase, whereas in the later stages, the gastric secretions essentially halt. Okay, so that is due to the inhibitory control by the secretin, cholecystokinin, and somatostatin, GIP, VIP, etc. It will lead to the inhibition and ceases the secretion of gastric juice. Okay, because the food has already reached the intestine, and hence gastric secretions are not necessary. Okay. Next question. The function of Meissner's and Meindrick plexus or Orbach's plexus in intestine is. The options are sympathetic, parasympathetic, sensory, both sympathetic and parasympathetic. The answer is both sympathetic and parasympathetic stimulation is occurring. Okay. So the digestive nerve plexus are intricate layers of nervous tissue that controls the movements in the esophagus, stomach, and the intestines. So two major nervous centers are there. One is the Meindrick plexus or the Orbach's plexus or the outer plexus. Okay. And next is the submucous plexus or the Meissner's plexus or inner plexus. Okay. First is the outer plexus, and next is the Meissner's or the inner plexus. Okay. So this is the basic organization of the gastrointestinal tract. So the outwardly we can see serosa or adventitia, and next layer is the muscularis propria. It has a outer longitudinal muscle and the inner circular muscle, and in between them we can see the Orbach's or the Meindrick plexus. This is the main nervous control in the intestine. And next layer is the submucosal layer, and in the submucosal layer there is Meissner's or the submucosal plexus. And the innermost layer is the mucosa containing epithelium, lamina propria, and muscularis mucosa. Okay. So innermost part is lined by the mucosa. So what is Meindrick plexus or Orbach's plexus? It's a part of enteric nervous system. Enteric means something related to the gastrointestinal tract, and existing between the longitudinal and circular layers of muscularis externa in the GIT. It is found in the muscles of esophagus, stomach, intestine, everything. Okay. And the Meindrick plexus, it originates from the medulla oblongata as a collection of neurons from the ventral part of the brainstem. And the vagus nerve then carries the axons to their destination in the GIT. Okay, they basically contain dogial cells. Dogial cells are present in both myendric as well as the mesenteric plexus. And it provides motor innervation to the both layers, that is the longitudinal as well as the circular muscles present in the GAT, having both parasympathetic and sympathetic input, controlling mainly the gastrointestinal movements. That's what the question was asking about. Okay. So, coming to the submucous plexus or the Meesner's plexus, they are also a subdivision of this myendric plexus only. Okay, it lies in the submucosa, the inner part of the intestinal wall, and is derived from the myendric plexus itself. And the branches from the myendric plexus, it perforates the circular muscle fibers to form the submucous plexus. Okay, from the myendric plexus, it perforates the inner layers, that the circular muscle fibers, to reach the submucosa to form the submucosal plexus. And the ganglia from the plexus extend into the muscularis mucosae and to the mucous membrane. They also contain dogial cells. And the nerve bundles of the submucous plexus, they are finer. Okay, from the when compared to the myendric plexus, these fibers are smaller or finer, and it controls mainly the gastrointestinal secretion as well as the local blood flow is controlled by the Meesner's plexus. Whereas this muscularis layer, they provides the motor innervation because they are located between the muscles, and hence they provide the motor innervation to the muscular layer of the gut. It has both parasympathetic as well as a sympathetic input. Okay, what will do the sympathetic response will lead to loss of secretion or reduced secretions, and muscular activity also reduced by the parasympathetic input, and the parasympathetic input will provide increase in secretions as well as the muscular movements in GAT, peristalsis, everything will be promoted by the parasympathetic or the normal pathway system. Okay. So this is the location. Here, between the outer longitudinal muscle and inner circular muscles, we can see the myendric plexus. And by piercing the circular muscles, it enters the submucosa, that is called Meesner's plexus. They are somewhat finer when compared to the myendric plexus. Okay. Next is Peyer's patches is seen. Peyer's patches are seen in, the options are ileum, duodenum, jejunum, stomach. So the answer here is ileum or ileum, whatever. Okay, so what are Peyer's patches? They are lymphoid tissues. Okay, they're basically small mass of lymphatic tissue which are found throughout the ileum, the region of the small intestine. Okay, so it's a part of small intestine, no? So this is a picture demonstrating the Peyer's patch, which contains this lymphoid organs, which is present in the ileum region of the small intestine. So coming to the next question, all of the following about secretin is false except. The options are it's secreted from the pancreas, it's secreted from the small intestine, it increases the insulin secretion, it's maintained by cholecystokinin. What's the answer? So the answer here is it is secreted from the S cells of the small intestine. Okay, so it is secreted by the S cells which are located deep in the glands of the mucosa of the upper portion of the small intestine, and its action on the pancreatic duct cells is mediated by cyclic AMP. It promotes the pancreatic secretions, causing a secretion of watery alkaline pancreatic juice. Okay, and the secretin increases secretion of bicarbonate by the duct cells of the pancreas and the biliary tract. It causes a secretion of watery alkaline pancreatic juice. Okay, it also decreases. It has an inhibitory function on the gastric acid secretion, and it may cause the contraction of the pyloric sphincter. Okay, contracts the pyloric sphincter in order to prevent the backflow of bolus. Next is the respiratory system. So these are some of the high topics in relation to respiratory system. That's you have to learn in detail about the lung volumes and capacities, respiratory pressures. The transport of carbon dioxide and oxygen is very important. Hypoxia. Okay, that's a shared topic that I have mentioned in the previous session also. Oxygen hemoglobin dissociation curve, muscles of respiration, regulation of respiration. Okay, these are the important high yield topics in relation to respiratory system. Okay, now coming to the questions. First question is, during inspiration, when the diaphragm contracts, the intrapleural pressure becomes. The options are more negative, less negative, positive, equal to intra-alveolar pressure. So what's the answer here? Answer is more negative. Okay, more negative. More negative means if it is positive, what happens is the pressure is much high and the air will not further enter the lung. Okay, so it is maintained to be negative. So the intrapleural pressure is always and always negative due to the atmosphere. When compared to the atmospheric pressure, it is always negative. During inspiration, it is minus 6 millimeter of mercury, and during expiration, it reduces to minus 2 millimeter of mercury. So intrapleural pressure that prevents the collapse of lungs. Okay, when the pressure inside is low, then the air enters the lungs. Okay, in order to do that, during inspiration it is minus 6, whereas in the expiration it becomes minus 2. And the intra-alveolar pressure is minus 4 millimeter during the millimeter of mercury during inspiration and plus 4 during expiration in order to propel the air outside. Okay, move the air outside, it becomes positive during expiration. And it helps in the flow of air in and out of the alveoli and helps in the exchange of gases. Okay, so what is called transpulmonary pressure? The difference between the pleural pressure and the alveolar pressure is called transpulmonary pressure. And the larger the transpulmonary pressure, the more the alveoli and the lungs will be inflated at any given moment. Okay. So transpulmonary pressure should be maintained in order to keep the alveoli as well as the lung inflated. Okay. So what happens during an inspiration? This volume of the lung will increase, that will release the pressure, and the pressure decreases and the air will flow in. Okay. And when the volume decreases during the expiration, the pressure increases greater than the atmosphere and the air will flow out. Okay. So the pleural pressure is always more negative to create the negative pressure vacuum for the inflation. And transpulmonary pressure, that's the larger the transpulmonary pressure, the greater the inflation of the alveoli and the lungs. Okay. So this is the transpulmonary pressure and this is the intrapleural pressure. Sorry, intraalveolar pressure, and the difference is what we call as transpulmonary pressure. This is the intraalveolar pressure and this is the intrapleural pressure. Intrapleural pressure should be less than intraalveolar. Why? If this is more, then it will make the alveoli constricted. It will press the alveoli. Okay, that's why intraalveolar pressure is maintained more than the intrapleural pressure. Okay. So coming to the next question, a lecturer took class to the students about the respiratory function. He asked students to analyze few cases. They analyzed respiratory function in 100 patients. Which of the following would best describe the restricted lung disease? So they're asking about the restricted lung disease and they are telling some of the stories. Okay, no need to read such stories. You have to first read the last sentence. In case of this type of large questions, you have to read the last sentence and after that, proceed with the other sentences. Okay. In the last sentence, usually they may be asking the question which they are meant to be asked. And the options are increase in residual volume, a decrease in total lung capacity. Third option is the decrease in tidal volume and increase in FEV1 by FVC. Forced expiratory volume in 1 second by forced vital capacity. Okay. That is FEV1 by FVC. Okay. So the answer here is, they're basically asking me about one thing. What is the quality of this restricted? What's the thing that's happening in the restricted lung disease? That's what they are asking. Okay. So the answer here is decrease in total lung capacity. So let's learn in brief about the restrictive and obstructive type of lung diseases. So lung pathologies, they can be basically divided into obstructive as well as restrictive type of lung diseases. In obstructive lung disease, what happens is that it makes the patient hard to exhale all the air in the lungs. Okay, so air is not coming outward. Some obstruction is there to prevent the coming out of the air or the exhalation of the air. And restrictive lung disease means something is restricting the lungs to expand fully. That is causing reduction in the air that's entering the lungs, or the expansion of the lung by the air is prevented in restrictive type of diseases. Okay, so what are the peculiarities happening in both disease? First is the total lung capacity. In obstructive, what happens is normal to increase. Why normal to increase? Because the air is not going outside. i.e. the exhalation is somewhat limited in case of obstructive diseases. Whereas in restrictive diseases, inhalation is decreased and hence the total lung capacity should decrease. Okay. Next is the residual volume. Here, obstructive lung disease, what happens is residual volume will increase. Why? The patient cannot exhale properly, so more amount of air is residing in the lungs. So, residual volume is increased in case of obstructive disease, whereas in restrictive, what happens is it will decrease. And the vital capacity in both, they are both affecting the lungs, and hence the vital capacity is decreased in both the type of diseases. Next is forced expiratory volume in 1 second divided by forced vital capacity. That will decrease obstructive. Why? Because expiratory volume will be reduced. Just a minute. Okay, let's continue. So the expiration is reduced and hence FVB and vital capacity is also reduced. But here in case of obstructive, FEV1 is also reduced and hence it is decreased in obstructive. Whereas in restrictive, what happens is FEV1 is not affected, whereas FVC is reduced, and hence in restrictive type, it is either normal or increased. And diffusion capacity is essentially normal in obstructive type of diseases, whereas in emphysema it reduces. Okay, and in restrictive it will decrease. And lung compliance, it is unaffected in case of obstructive, whereas it is increased in emphysema. Okay, emphysema is a exception. Okay, so restrictive, what happens is compliance is decreased. So compliance means the loss of elasticity. Okay, compliance is increased when the loss of elasticity is lost by the loss of elasticity of the lung happens. So the hallmark, it's a hallmark of obstructive pattern of disease is the decreased expiratory flow. That's the definition says. Okay. The hallmark of restrictive lung disease is that a decrease in the total lung capacity. This can also be asked as a future question. Also, total lung capacity, lung compliance or the lung elasticity and vital capacity is reduced in case of restrictive type of lung diseases. Which of the following is used to measure anatomical dead space? So, anatomical dead space means what? Okay, first explain the question. The options are inert nitrogen sampling, capnography, spirometry, Bohr's method. What's the method used to determine the anatomical dead space? The answer is inert nitrogen sampling. Okay. So what is basically anatomical dead space? That means the part of the respiratory tract which are not involved in gaseous exchange. Okay, which are not involved in gaseous exchange. That means nose, larynx, etc. They are only acting as a passageway to for the air movement, and not any gaseous exchange is not taking place in such parts of the respiratory tract, and hence they are called anatomical dead space. Okay, so there is a physiological dead space also that is involving the alveoli. Okay, in some diseases, this alveoli, some of the alveoli are not involved in the gaseous exchange, and also in some cases, what happens is alveoli is not diseased, but the amount of blood supply or the ischemia happening to the alveoli is preventing from doing the gaseous exchange. And when this two things are also added to the anatomical dead space, that is called physiological dead space. Okay. So anatomical dead space measures the volume of all the dead space of the respiratory system other than the alveoli and their other closely related gas exchange areas. Okay. Whereas physiological means this alveolar dead space is also included along with the anatomical dead space. And usually this anatomical dead space, it's measured with single breath nitrogen method or the Flowers method. And in physiological dead space is measured by Bohr's method of equation. Okay. So in normal person, this anatomical as well as physiological dead space, both are one and same in normal person. Why? This alveoli is not involved in a normal person. Okay, in diseased persons only this alveoli is involved in, not involved in gaseous exchange. So in normal person, its anatomical and physiological dead spaces are essentially the same. So the normal dead space in dead space of air in young adult man is about 150 milliliters. Okay. So basically this anatomical dead space, it's a volume of respiratory tract from nose up to terminal bronchiole, because these are areas are not involved in gaseous exchange. Instead, they are acting as a passageway for the air. So coming to next question, carbon dioxide is carried in the blood. The options are in combination with hemoglobin, in physical solution in plasma, in combination with the plasma proteins. All the above. What's the answer here? All the above. Why? Because these are all different mechanisms of transport of carbon dioxide in blood. Okay, it can combine with the hemoglobin. It can combine as a physical solution in plasma. Okay, it can combine as the plasma proteins to form carbaminohemoglobin. Okay, and get transported. So transport of carbon dioxide in blood occurs by three main methods. It can dissolve in plasma. It can bound to hemoglobin and as bicarbonate ions. And 7% of carbon dioxide, it travels in the blood, dissolved directly in the blood plasma. And 70% is carried in blood as bicarbonate ions. There is a mechanism called chloride shift or Hamburger shift. Okay. And the carbonic acid, it breaks down into bicarbonate ions as well as hydrogen ions. And usually these bicarbonate ions then leave the RBCs and okay, I will explain using this diagram. Initially, what happens is in tissues, there is a lot of carbon dioxide. Okay. So this carbon dioxide that will enter the RBC. Okay. And inside the RBC, there is an enzyme called carbonic anhydrase that will promote the combination of carbon dioxide with the water forming carbonic acid, H2CO3. And this carbonic acid is very unstable and hence it is split into bicarbonate ion and hydrogen ion. Okay. Inside the RBCs. And why this carbon dioxide is not combining with the water outside the RBC? Because of the presence of carbonic anhydrase inside the RBC, about 100 to 200 times more formation of carbonic acid is done inside the RBCs compared to the plasma. Okay, so here the carbon dioxide enters the RBC and it combines with the water inside the RBC forming carbonic acid, and it is splitting into bicarbonate ions as well as the hydrogen ions. What happens is the bicarbonate ions is coming outside to the concentration gradient, and in order to maintain the electric neutrality, what happens is chloride ions is entering inside. And this is happening in the band 3 of the RBC membrane. It acts as an antipode by extruding the bicarbonate ion outside the RBCs and including or internalizing the chloride ions from the outside. So outside plasma, we have a lot of sodium chloride, and sodium chloride is present as sodium ions as well as chloride ions. So in order to maintain the electrical neutrality, this minus ion is coming, and hence to substitute that, this chloride ion is entering the RBC. And hence this bicarbonate, it combines with the sodium ions outside the seen in the plasma and forms the sodium bicarbonate, and it is transported to the lung alveoli for its elimination. Okay, and inside this is what we call as chloride shift, because in order to substitute for the bicarbonate ion, the chloride is entering by means of band 3. In the band 3 of RBC membrane, there is a band 3 protein. It acts as an antipode. Okay. And this is what we call as chloride shift or Hamburger phenomenon. There is a reverse chloride shift. This occurs in the alveolus. In alveolus, what happens is this oxygenated hemoglobin is there. There is a lot of oxygen in the alveolus. And hence what happens is bicarbonate ions is moved outside. And chloride ion is, the outside bicarbonate ion is split from the sodium bicarbonate into sodium ions and bicarbonate ions. That is in the plasma. Okay. And from the plasma, this bicarbonate ion is entering the alveolus. This is occurring in the lung alveoli. Okay. Inside the RBC of the lung alveoli, this bicarbonate ions are entering, and to substitute that, this chloride ions are moved out. Okay. And also there is H plus ion present in the hemoglobin, and it will combine to form carbonic acid, and it will split to carbon dioxide and water, and it will transport it and is eliminated from the lung system or the alveolus. Okay, this is what we call as reverse chloride shift. So swapping of bicarbonate and chloride ions is called chloride or Hamburger shift. So coming to the next question, the major sign of hyperventilation is. So we have to notice whether they are asking sign or symptom. Okay. So the options are cyanosis, dyspnea, hypercapnia, hypoxia. What's the major sign of hypoventilation? So the answer here is hypocapnia. Hypoventilation means the carbon dioxide is retained. It's not eliminated because of the hypoventilation. So carbon dioxide increases and causes hypercapnia that will promote the respiratory centers causing dyspnea. That's a symptom. Okay. So in hypoventilation, the amount of air moving in and out of lungs is reduced. So carbon dioxide is retained and that will lead to hypercapnia. Okay. So this hypercapnia, that's a sign. It stimulates the respiratory center excessively and it will lead to dyspnea or difficulty in breathing. And so hypoventilation can result in hypoxia, hypercapnia, dyspnea, and finally death. Okay. So another question relating to this is, which of the following does not stimulate the alveolar hyperventilation? The options are hypoxia, hypercapnia, acidosis, stretching of airways. So hypoxia, hypercapnia, these are all. This can promote hyperventilation. Okay. This is the result of hyperventilation and hence they lead to hyperventilation. Okay, by reflex pathways, by the stimulation of the respiratory system. Acidosis also same, but stretching of airways is not a factor leading to hyperventilation. So the answer here is stretching of airways. Next question. Increase in the partial pressure of carbon dioxide leading to release of oxygen to the tissues is. So partial pressure of carbon dioxide is high, leading to release of in tissues. Usually carbon dioxide amount of pressure is high and it can lead to release of oxygen into the tissues. So this effect is called, it's the Bohr effect. Okay. So the presence of carbon dioxide decreases the hemoglobin affinity. The presence of carbon dioxide, it decreases the affinity of hemoglobin for oxygen and enhances the release of oxygen to the tissues and shifting the oxygen dissociation curve to right, towards the right. That's what we call as Bohr effect. Okay. And next is the combination of more amount of oxygen with hemoglobin, it displaces the carbon dioxide from hemoglobin. Okay. When more amount of oxygen is available, it displaces the carbon dioxide from the hemoglobin and the excess of oxygen concentration resulting in the shift of carbon dioxide dissociation curve to right. Okay. More carbon dioxide is displaced from the hemoglobin in the presence of oxygen. That's what we call as Haldane effect. Okay. So what happens in Haldane effect? The carbon dioxide. You have to notice whether they are asking about oxygen dissociation or carbon dioxide dissociation. Okay, here they are asking about carbon dioxide dissociation curve. In Haldane effect, what happens is carbon dioxide is released and hence the carbon dioxide dissociation curve was shifted to right. Okay. Next question. The limitation of inspiration by vagal lung inflation signals is called. The options are autonomic reflex, Bainbridge reflex, Herring-Breuer reflex, dynamic stretch reflex. What's the answer? It's the Herring-Breuer reflex. Okay. So it's a stretch reflex. Okay. It is due to some stretch receptors which are present on the walls of bronchi and bronchioles of lungs. During inspiration, when there is a stretching of lung tissues due to expansion, and the stretch receptors are stimulated and they produce impulses which are carried by the vagal efferent fibers to the respiratory centers. Okay, there is a stretch that is due to increased inspiration and hence this stretch is noted by the stretch fibers and it is carried to the respiratory centers. And Herring-Breuer reflex inhibits the inspiratory center and hence it will stop the inspiration and stimulates the expiration. What is this for? It's a protective reflex which limits the overstretching of lung tissues. Beyond a point, we cannot inspire more amount of air because of this stretch receptors, and this particular phenomenon is called Herring-Breuer reflex. Okay, it's a protective reflex which limits the overstretching of lung tissues. Another stretch reflex is there in the cardiovascular system, that is called Bainbridge reflex. Okay, so the increase in heart rate secondary to an increase in the central blood volume. That's due to the stretch receptors in right atrium. That's what we call as Bainbridge reflex. Okay. So next is Kussmaul's breathing. Kussmaul's respiration occurs in response to. The options are decrease in pH of blood, increase in pH of blood, obstructive pulmonary disease, carbon monoxide poisoning. Usually, one trick in solving MCQs is that if there is a decrease and opposite things are there in the option, i.e., in here, we can see increase in pH or decrease in pH is there. So, one of them may be the answer. Okay. Usually, this options, one of these options may be the answer. You can eliminate other options. Okay. So, that's another trick in solving the MCQs. So, here the answer is decrease in pH, i.e. acidity. Acidosis will lead to Kussmaul's respiration. Okay. So decrease in pH of blood or severe acidosis increases the rate and depth of respiration. That's what we call as Kussmaul's breathing. Next question. Oxygen dissociation curve. This is also an important topic. Many questions are asked regarding this topic. Okay. So oxygen dissociation curve shifts to the right by all of the following except. Usually this oxygen dissociation curve or carbon dioxide dissociation curve, either of them, any of them, what happens is shift to left means affinity to the particular thing. Either it is carbon dioxide or it may be oxygen. What thing they are, it's based on what they are asking. Okay. So in dissociation curves, shift to right means least affinity and shift to left means more affinity. Okay. So here they are asking about oxygen dissociation curve. It shifts to right by all of the following except, okay, increase in temperature, increase in carbon dioxide tension, rise in pH, rise in H plus ion. These are opposite options. Okay. So any of them may be the answer. Okay. Rise in pH means alkalinity. Rise in H plus ion means acidity or dip in pH. Okay. So the answer here is rise in pH. A shift to the left indicates the acceptance of oxygen. I have already mentioned that the shift in left means acceptance of oxygen by hemoglobin, and shift to right indicates dissociation of hemoglobin, oxygen from hemoglobin. Okay. So these are the basic factors favoring the shift, right shift. That is decreased partial pressure of oxygen, increased partial pressure of carbon dioxide. That's what we already mentioned. That is the Bohr effect. Increase in H plus ion concentration of decreased pH. Excess in 2,3 dpg. 2,3 dpg levels means oxygen will be released from the hemoglobin more readily. Okay. Usually during exercise and high altitudes, increased body temperature, etc., there is an excess of 2,3 dpg. Okay. So this is the oxygen hemoglobin saturation dissociation curve. Okay. Shift to right means what? Reduced affinity, oxygen is more readily released. Shift to left means oxygen affinity is increasing and oxygen will not be released. Okay. So these are the various factors favoring shift to right that is already mentioned. And shift to left is decrease PCO2. That's the Haldane effect, and decrease H plus concentration or increase pH. So that's alkalinity, decrease 2,3 dpg, decrease body temperature. Okay. These are the basic factors affecting the oxygen hemoglobin dissociation curve. So next question is, within the RBC, hypoxia stimulates glycolysis by which of the following regulating pathways? The options are hypoxia stimulates pyruvate dehydrogenase by increased 2,3 dpg. Second is hypoxia inhibits hexokinase. Hypoxia stimulates the release of all glycolytic enzymes from the band 3 on RBC membrane. Fourth option is activation of regulatory enzymes by high pH. What's the answer here? So hypoxia stimulates the release of all glycolytic enzymes from the band 3 on the RBC membrane. Okay. That will lead to increased glycolysis and leading to the production of 2,3 dpg. So it causes deoxygenation of the hemoglobin. Okay, hypoxia basically causes deoxygenation of the hemoglobin and increases the band 3 tyrosine phosphorylation and thereby stimulate the glycolysis by releasing glycolytic enzymes from the band 3 on the RBC membrane. Previously also we have mentioned about that antiport action of band 3 in chloride shift or Hamburger phenomenon. Okay, that also you can remember along. So next question is, oxygen acts as an emergency drug. Oxygen as an emergency drug is contraindicated. First, the options are anaphylaxis, asthma, hyperventilation, COPD. So what's the answer here? Hyperventilation. Why hyperventilation? In hyperventilation, oxygen is contraindicated because it causes hyperventilation causes hypocapnia, that is decrease in the carbon dioxide levels. So it will lead to the narrowing of blood vessels that supply the blood to the brain. Okay. So what happens is hypocapnia, that means less amount of carbon dioxide. That is interpreted by the higher centers as ample amount of oxygen receiving. Okay. So that is lesser amount of carbon dioxide means adequate oxygen is reaching the brain. That's what they are assuming. Okay. And hence they lead to the narrowing of blood vessels that supply blood to the brain. So if oxygen is administered, that means further reduction in carbon dioxide occurs. So that will again shrink the already narrowed blood vessels that are supplying the brain. So we are not administering oxygen in case of hyperventilation. Okay, that's the reason. So next question is, in hyperbaric oxygen therapy, oxygen is given as. So the options are 10 dives at 100% saturation, 50 dives, 30 dives, 40 dives. It's just a memorizing type of question. Okay, that is 30 dives in 100% saturation. It's the answer. So usually in HBO, we prescribe 20 to 30 dives at 100% oxygen and 2 to 2.5 atmosphere pressure. That you have to learn by heart. Okay. Next is severe OSA is diagnosed by apnea-hypopnea index score. Okay, this is also that memorizing type of question. The options are 5 per hour, 15 per hour, 25 per hour, and more than 30 per hour. Severe is more than 30 per hour. Okay. So apnea-hypopnea index or AHI, it indicates the severity of sleep apnea. Okay. It is represented by the number of apnea or hypopnea events per hour of sleep. Okay. It's calculated per hour of sleep and it is represented by the number of apnea and hypopnea events. And apneas, that means pauses in breathing. Okay. It must last for at least 10 seconds. It should be associated with decrease in blood oxygenation also. Okay. So it must last for at least 10 seconds and it should be associated with decrease in blood oxygenation or saturation levels. And comparing AHI and oxygen desaturation, it gives an overall sleep apnea severity score that evaluates both the number of sleep disruptions and the degree of oxygen desaturation should be assessed. I.e., we have to assess the sleep disruptions caused by apneas or hypopneas. And AHI is usually calculated by dividing the number of apnea events by the number of hours of sleep. Okay, number of apnea events occurring in different number of hours of sleep. It is divided by number of hours of sleep. Okay, so there is a chart regarding that. Normal AHI values is usually less than 5 and mild sleep apnea, that is 5 to 15, denotes mild sleep apnea, whereas it is 15 to 30, it is moderate, and severe, it's above 40, above 30. Okay. So coming to the next question. At rest, which muscles help in respiration? The options are mainly diaphragm, internal intercostal muscles, external intercostal muscles, mainly diaphragm and external intercostal muscles. So what's the answer here? So mainly diaphragm. Okay. So what are the muscles involved in respiration? First is the diaphragm and the intercostal muscles. Both external as well as the internal intercostals are involved in respiration. Also the axillary muscles are sternocleidomastoid, anterior serrate muscles, scalene muscles, and abdominal rectus muscles. Okay. So what are the muscles involved in different situations? In a normal quiet breathing, both inspiration and expiration, only diaphragm is involved. Okay, in a normal quiet breathing, only diaphragm is involved. And heavy breathing, like in exercise, when the muscles are elevated and the chest cage come into action during inspiration, during exercise, these are the muscles. That is external intercostals, sternocleidomastoid muscles, anterior serrati, and scalene muscles. And in heavy breathing, like in exercise, when the expiration is done, then these muscles, that is the abdominal recti and internal intercostals, are involved in expiration during the heavy exercise. Okay. Next question is, end-tidal carbon dioxide is decreased in all except. The options are cardiac arrest, hypotension, hypothermia, pulmonary embolism. So the answer here is hypothermia. Okay. So we are going to discuss about the factors which are causing the decrease and increase in end-tidal carbon dioxide. Okay, what is end-tidal carbon dioxide? It's a non-invasive technique which measures the partial pressure or maximal concentration of the carbon dioxide at the end of an exhaled breath. I.e., after the end of an exhaled breath, the amount of carbon dioxide remaining is measured or monitored. That's what we call as end-tidal carbon dioxide monitoring. Okay, it is expressed as a percentage of carbon dioxide or millimeters of mercury. And the normal values are usually 5 to 6%, which is equivalent to 35 to 45 millimeters of mercury. Okay. So these are the factors causing the changes in partial pressure of end-tidal carbon dioxide. So increase can be caused by these factors. That is the increased carbon dioxide production and delivery to lungs. That is i.e. due to increased metabolic rate, fever, sepsis, etc. And decreased alveolar ventilation. That is due to hypoventilation, respiratory center depression, partial muscular paralysis, and things like that. And equipment malfunction can also lead to increase in end-tidal carbon dioxide. That is in rebreathing, exhausted carbon dioxide absorber, leak in ventilator circuit, etc. And decrease can be due to decreased carbon dioxide production and delivery to the lungs, caused by hypothermia, pulmonary hypoperfusion, cardiac arrest, etc. And increased alveolar ventilation is seen as such in hypoventilation, and equipment malfunction like ventilators disconnected, esophageal intubation, complete airway obstruction. Okay, like that. Next is lung compliance is increased. The options are surfactant deficiency, pulmonary fibrosis, emphysema, acute pulmonary edema. Answer is emphysema. Okay. So, what are the factors? What is compliance and what are the factors leading to increase and decrease in compliance? Okay. So, increase in compliance. Compliance increase usually due to the loss of elastic property of lung tissues. Okay. Loss of elastic property. So it can be physiological as well as pathological. Physiologically, it usually occurs in the old age, and pathological condition is mainly emphysema. Okay. And alveolar septa, which provides some of the retractive forces in lungs, are destroyed in both conditions. In emphysema, what happens is multiple alveoli is destructed and holes are made in the lungs due to disease. And this alveolar septa, it provides some of the retractive forces. The elasticity of the lungs is provided by the alveolar septa, and they are destroyed in case of physiological old age. Some amount of destruction occurs in during aging, and most of the destruction occurs in diseased conditions like emphysema. Okay, extensive loss of septa can be due to emphysema. And emphysema, it increases the compliance secondary to loss of normal elastic recoil of lungs. And normal to moderate obstructive or reactive airway disease increases the compliance. Okay, obstructive means failure of proper exhalation is not possible in obstructive diseases. So, this is the variations in lung compliance. In fibrosis, this compliance is decreased. Lung cannot expand properly. Okay, something is restricting the expansion of the lungs. So compliance is decreased in fibrosis, whereas it is increased in emphysema. Okay. So what are the factors leading to decrease in compliance? As I have already mentioned, restrictive diseases or fibrosis can cause decrease in compliance. So it decreases in pathological conditions that produce scarring or fibrosis of lungs, such as deformities of thorax like kyphosis, scoliosis, etc., paralysis of the respiratory muscles, pleural effusion, abnormal thorax such as pneumothorax, hydrothorax, etc., and in diseases like TB, silicosis. So reduced compliances produce a condition termed restrictive lung disease, and these patients, they have to generate greater forces for the expansion of lungs. Okay, they have to exert more force for the expansion of lungs. Coming to the next question, this is a recent question. Which of the following is a feature of surfactant? The options are produced by type 1 pneumocytes, helps prevent the collapse of alveoli, increase in volume of alveoli, helps in the collapse of alveoli. You can, by reading the option itself, you can eliminate the fourth option. Okay. And the other three options are there. So the answer here is, what's the feature? It prevents the collapse of alveoli. That is what surfactant is for. Okay. They are produced by the type 2 pneumocytes. Okay. So it lowers the surface tension of the fluid lining the alveoli and prevents the collapsing tendency of the lungs by reducing the surface tension. It is a phospholipid, and usually the absence of surfactant results in hyaline membrane disease or respiratory distress syndrome. Okay, and it is usually produced by the type 2 pneumocytes. So coming to the next system, that's a renal system. So these are some of the high-yield topics of renal system. GFR and the factors affecting it. Countercurrent mechanism is important. Juxtaglomerular apparatus. Renin-aldosterone angiotensin system. Okay, these are the important topics. Coming to the first question. In osmotic diuresis, the options are increase in renal blood flow, increase in glomerular filtration rate, increase in sodium chloride concentration in urine, decrease in reabsorption of sodium chloride. So what happens in diuresis? It's the increase in sodium chloride, and diuresis means increase amount of water and solutes in urine. Okay, so decrease reabsorption of sodium chloride. Decrease reabsorption of sodium chloride. That's what we call as diuresis. Not only sodium chloride, water is also not reabsorbed and hence leading to diuresis. Okay. So some substance, it increases the osmotic pressure in the tubular fluid. Increasing the osmotic pressure in the tubular fluid leads to attraction of water and sodium chloride inside the tubule, and that will lead to excess of water and solutes through the urine via osmotic diuresis. Okay, by altering the osmolarity of the tubular fluid, increased excretion of water and solutes can occur. That's what we call as osmotic diuresis. Okay, so mannitol, urea, sucrose, glucose, they are best osmotic diuretics. Okay. So next question is, brush border is seen in. The options are Bowman's capsule, PCT, DCT, loop of Henle. So what's the answer here? It's a PCT. Usually we can see brush border cells in the PCT. The brush border cells are seen in the PCT and hairpin band is seen in loop of Henle. It's understood. Okay. So this is the histological image depicting the brush border epithelium. Okay, it's in the PCT. Next question. Tubuloglomerular feedback occurs in. The options are AD secretion collecting duct, constant solute load on distal tubule, macula densa, balance between ascending and descending loop of Henle. So the answer here is constant solute load to distal tubule. It's a feedback mechanism, no? So what is tubuloglomerular feedback? As the rate of flow through the ascending limb of loop of Henle and the first part of the distal tubule increases. That means the distal parts of the nephron, they are showing increased rate of flow. Okay, so glomerular filtration in the same nephron decreases, and conversely a decrease in flow increases. That means the rate of flow is increased in the distal part of the nephron. That means more filtration is happening. So as a feedback mechanism, there will be a decrease in GFR. Okay. And this is called the feedback. The flow is increased in the distal parts. Okay. That indicates a higher GFR. So as a feedback mechanism, GFR will be reduced. That is what we call as tubuloglomerular feedback. And it maintains the constancy of load delivered to the distal tubule. Okay. And next is the glomerulotubular balance. An increase in GFR, it causes an increase in the reabsorption of solutes. Okay. When GFR is increased, what happens is reabsorption will be increased. So and consequently, water primarily in the proximal tubule is reabsorbed more. So that generally the percentage of solute reabsorbed is held constant. Okay. If say 100x is filtrating, and then according to that, the percentage of the solute and the water reabsorbed is also increased. So that means a percentage of solute reabsorbed is held constant. Okay. That is what we call as glomerulotubular balance, and it's particularly prominent for sodium ions. Okay. So next is, water is mostly absorbed in which part of the nephron? Collecting duct, PCT, DCT, loop of Henle. It's the DCT. Okay. So about 65% of the filtered load of sodium and water and the slightly lower percentage of filtered chloride. Mainly this reabsorption is happening in the PCT, okay, before the filtrate reaches the loop of Henle. And the water absorption also, it occurs by the osmotic diffusion. That is the osmotic gradient maintained in the interstitium and the tubule. That's responsible for the reabsorptions. Next question. In the presence of excessive ADH, which part of the nephron shows hypertonic? Which part shows maximum hypertonic fluid? The options are distal, proximal, loop of Henle, collecting tubule. So the answer is collecting tubule. Okay, so ADH mainly acts on the collecting tubule. And ADH binds to the receptors on the cell in the collecting duct of the kidney, and it promotes the reabsorption of water back to the circulation. Hence, the urine becomes hypertonic. Water is reabsorbed and the urine is concentrated and it becomes hypertonic by the action of ADH on the receptors that are present in the collecting duct. And in the absence of ADH, the collecting ducts are usually they are not permeable to water. Okay, and hence the water is not reabsorbed from the collecting duct. Only a minor amount of water is reabsorbed from the collecting duct. Okay, so only minor amount of diuresis, amount is differed by the action of ADH. So in the absence of ADH, the collecting ducts, they are mainly impermeable to water, and hence this water is flowing as urine. And antidiuretic hormone, it stimulates the water reabsorption by stimulating the insertion of some water channels are inserted into the membranes of the kidney tubules in the collecting ducts. And these channels, they transport solute-free water. Only water is reabsorbed. No solutes are reabsorbed from the collecting duct. Okay, through the tubular cells and back into the blood, leading to decrease in the plasma osmolarity and increase in osmolarity of urine. Okay, so urine is hypertonic after the action of ADH on the collecting duct. Next question. The macula densa is formed from the cells of. The options are efferent arteriole, ascending limb of loop of Henle, DCT, collecting duct. The answer is DCT. Okay, distal convoluted tubule. So, the first portion of the distal tubule usually forms the macula densa. It's a group of closely packed epithelial cells that's a part of juxtaglomerular complex and provides feedback control of GFR and blood flow in this same nephron. Okay, so it provides this juxtaglomerular apparatus. It provides the feedback control. Okay, the macula densa, the neighboring Lacey cells and the renin secreting JGS cells in the afferent arteriole, they form the juxtaglomerular apparatus. So what's the composition? Some macula densa, neighboring Lacey cells and the renin secreting juxtaglomerular cells that is present in the afferent arteriole. That forms the juxtaglomerular apparatus. Okay. So it's the image showing macula densa. Next question, aldosterone axon. The options are DCT, PCT, collecting duct, loop of Henle. So what's the site of action of aldosterone? It's the collecting duct. Like the ADH, the site of action of aldosterone is also collecting duct. So aldosterone is an important regulation of sodium reabsorption and potassium secretion. Okay, it will absorb the sodium and secrete potassium into the renal tubules. And the primary site of aldosterone action is on the principal cells of the cortical collecting tubule. And what's the mechanism of action? It increases the sodium reabsorption while at the same time increases potassium secretion. It is by the stimulation of sodium potassium ATPase formed on the basolateral side of the cortical collecting tubule membrane. And it increases the sodium permeability in the luminal side. Okay, it increases the sodium permeability in the luminal aspect of the membrane. And both the late distal tubule and the cortical collecting tubule segments, it reabsorbs as a sodium ion, and the rate of reabsorption is controlled by different hormones, especially the aldosterone. Okay. So, what is Addison's disease? It results from the decreased secretion of the hormone. In hormone aldosterone, it impairs the ability of kidney to reabsorb sodium and leading to mild or modest degree of hyponatremia is sodium ion deficiency. Okay. So Addison's disease is due to the decreased secretion of aldosterone. Next question, that's a recent question. Which part of the nephron is responsible for the secretion of uromodulin or TAM horse valve protein? So the options are ECT, DCT, ascending loop of Henle, descending loop of Henle. What's the answer here? Ascending loop of Henle. So uromodulin or Tam-Horsfall protein is a glycoprotein. It's produced by the kidney tubular cells of the tubular cells of the ascending limb of loop of Henle. The thick portion of the ascending limb of loop of Henle cells, they produce uromodulin or Tam-Horsfall protein. Okay. Also, some amount is produced by the early distal tubules also. Coming to the next question. This is a very important question. You have to learn by heart, but what are the factors affecting the GFR? Okay, that you have to learn by heart. Not by heart, by using some logics, you can learn it very easily. Okay, so 22 years old healthy patient is enrolled for a drug trial evaluating renal perfusion. The drug causes increase in GFR and filtration fraction. So which of this is mechanism of action of this drug? Basically, they are asking the factors which are causing increase in GFR and filtration factors. Okay. So what are the options? Afferent arteriolar constriction, afferent arteriolar dilation, efferent arteriolar constriction, efferent arteriolar dilation. What's the answer here? So the answer here is efferent arteriolar constriction. Okay. Efferent arteriolar constriction means outflow is reduced and hence the filtration will be increased and the GFR will be increased. Okay. So this may be the drug mechanism of action of this particular drug. So constriction of the afferent arteriole. Constriction of afferent arteriole, it will reduce the amount of blood reaching the glomerulus and hence it will reduce the GFR, whereas constriction of the efferent or the outflow system, that will increase the GFR. Okay. There are many other factors which are affecting the GFR. So this is all about this topics. So some questions, they are very high yielding questions I have mentioned. There are also some amount of questions I have mentioned the high yield topics also. You can learn from that. Okay. So thank you for your patience and listening. Thank you. If you have any doubt, you can ask in the WhatsApp group. Okay. Thank you.